We do want to leave time for discussion. I think that's one of the most valuable parts of this. I'm just going to make a few, I'm not going to try and cover everything, of course, but I want to talk mostly focusing on just sort of what we know about checkpoint inhibitors, and then a little bit on the latest data on CAR T cells, and just make those points. So starting on checkpoint inhibitors, by now you're all familiar with, we have in control of T cell activation and growth, we have sort of negative effects. These are the brakes, the CTLA4 and PD-1, and we have the gas on the left side with lots of other positive regulators, which we don't have time to talk about some of these that are also very interesting. If you look at where these are working, the CTLA4 is a more sort of global priming phase activation of T cells, and it leads to, when you block that, you'll get a very strong polyclonal activation at a basic level. The PD-1, PD-L1 axis is more effective at at controlling immune responses physiologically in the periphery. So it's more of a fine-tuning of immune responses, and that's probably why we have fewer side effects, fewer autoimmune side effects when we use PD-1 blockade, whereas CTLA4 blockade leads to a much higher incidence and severity of autoimmune side effects. CTLA4 blockade can work in non-Hodgkin lymphoma. Steve Ansel and I did this study years ago and showed that, in fact, we saw with CTLA4 blockade alone, we saw responses in follicular lymphoma, and we even saw this a response in DLBCL, and Steve tells me that this patient is still in complete remission, hasn't relapsed now. I think it's after 7 or 8 years. So, but of course, what really captured our attention several years later was really the fact that this PD-L1 negative molecule, the regulator of T cells, was present on many, many different types of tumors and associated with poor prognosis, where the PD-L1 and PD-L2 ligands would bind to PD-1, inhibiting those T cells. And when a very important study by Yamamoto showed that this PD-L1 was highly expressed on Reed-Sternberg cells, and that also that inhibited T cells. And this was later shown by Margaret Shipp's group to be associated with the amplification of the 9p24 locus that includes PD-L1, PD-L2, and JAK2. And then we and others showed that this is PD-L1 was also expressed on both T cells and B cells of different histologic subtypes. And you can see here very strong expression on Reed-Sternberg cells, and some lymphomas, including some of these ones like Herbert just saw, transformed lymphomas. DLBCLs have extraordinarily high levels of PD-1 expression. Others are negative, but there may be a few macrophages that are PD-L1 positive in the tumor cell. But it's really this case where we think this is a much more important expression on the tumor cells. And in follicular lymphoma, you do see PD-L1 expression, but it has a different pattern. It's on these macrophages that are interdigitated between the follicles. And if you just look at sort of a whole mount of a follicular lymphoma, you can see the PD-L1 is between the follicles. When you go up close, you can actually see the PD-L1 positive macrophages between the follicles. And these are actually interacting with T cells that are shown here in pink. So once again, we've got different varying degrees. These kind of data led to, in 2012, the first phase 1 trial we did with nivolumab, one of the PD-1 inhibitors, in patients with all different types of B cell and T cell lymphomas and myeloma, and including Hodgkin lymphoma. And you're all familiar by now with the very striking responses that we saw in the first 23 patients of Hodgkin lymphoma, an 87% response rate. Almost every patient had a response here, and these responses were correlated with the presence of the PD-L1 and PD-L2 expression on the cells, and in all of 10 cases that we looked at, they had amplification of the PD-L1, PD-L2 locus, that 9p24 locus, and very high level expression by immunohistochemistry. And these responses were also durable, and that led us to also take on a phase 2 study. I'm sorry, I'm going to skip ahead to show that. Maybe we'll go. I'll make a point that these responses, as is seen here, can be very rapid. And some of these were some of the most striking responses I've sort of seen of any cancer. Here's a woman, actually a patient of Dr. Eridat's that probably should have flunked the entry criteria to come onto this trial. She had borderline PFTs because she had right upper lobe collapse and had a painful lesions in her pelvis. Like she couldn't even sit down on the exam table. And within 2 weeks, she came up and hopped up and sat down on the exam table and was breathing just fine. So this has told us right away that this was highly active. This is Steve Ansel's patient from the Mayo Clinic that was literally on their way to hospice when Steve called him and said the trial just opened, come on down, and this patient achieved. That's actually not a CR because there's a little stuff there, but that's a pretty darn good response. This patient's been in continuous remission as long as they've remained on nivolumab. So these data led to the phase 2 registration study that we completed, and here the response rate was not 87%, it was around 70%, but nonetheless, and this is the 205 study, led to the approval of nivolumab in patients who were double refractory to autologous stem cell transplant and brentuximab vedotin. And interestingly enough, as predicted, the level of amplification of the 9p24 locus was correlated with the degree of response. And you can see most patients still have a response, but if you have higher and higher copy numbers of the 9p24 locus, you're more likely to have a complete response here, as you can see in green. So there's, even though all the patients, virtually all of them have PD-L1 expression, the more you have, it seems to correlate with a better depth and even duration of response. So at the ASH meeting, we gave an update of some of these data and just show you briefly some of this. These are the nivolumab patients who are the double refractory. The median progression-free survival had been 8 months, but most of the patients had not been followed for very long at that point. Now the median progression-free survival for the whole group is 15 months, and those that are in CR are doing really well up here. And interestingly, even patients with stable disease and partial responses are doing well. And this is in contrast to brentuximab vedotin, where if you have stable disease or a PR, you tend not to have prolonged disease-free survival. So these responses tend to be pretty durable, but we do not yet know if there will be a plateau on these curves, and I think that's something we're going to have to wait several years to determine. Brentuximab naive seems to be the same situation, high response rate, 68%. And in here, since they haven't had that very important and active therapy, brentuximab vedotin, it seems like their responses may be a little bit better. They have more CRs, and their progression-free survival is, but this is once again an earlier cohort, so we'll have to wait and see. So how about nivolumab and checkpoint inhibitors in non-Hodgkin lymphoma? Well, in this trial, we did include follicular lymphomas and DLBCLs. And we did see a few responses, 30 to 40% in these subsets, but these are very small numbers. Interestingly, in these, there wasn't any response in mantle cell lymphoma or in SLL or marginal zone lymphoma, and it's known that these cells do not express PD-L1 on their surface. So it did seem to be enriched, the responses did seem to be enriched for the histologic subtypes that do have a proportion of their cases are positive for the PD-L1 inhibitor. PD-1 ligand, PD-L1. However, there were some responses that were durable in follicular lymphoma. The responses in DLBCL, they were mostly transient, although we did have a very striking response in one of our first patients that had recently had ibrutinib. However, that response only lasted about 6 months before that patient succumbed to their disease. So the bottom line is that these agents are active in and similar results are seen with the anti-PD-1 antibody pembrolizumab. So these agents are active against the non-Hodgkin lymphoma, but probably are going to find their most use in combination because they may not have really strong single-agent activity. Shown here is the PFS and OS curves here. Switching to CAR T cells, and we'll go through this quickly so that we have time. I think you're all familiar with the basic idea of removing T cells by leukapheresis, transducing them with retrovirus or lentivirus that encodes the chimeric antigen receptor, attacking CD19 and reinfusing those into the patient. Here is some data from the early NCI experience, and they saw some very striking responses in patients with large cell lymphoma. And I showed you this slide already, and it can respond in follicular lymphoma as well. And I wanted to just give an update of the study that was presented at the ASH meeting in the late-breaking session for those of you that didn't see it, and that is the KITE study. This is the vector that was licensed from the NCI, so this is sort of an extension of the NCI study. And what's important about the study is that they appear to be furthest along and closest to regulatory approval. And indeed, this is a breakthrough status accelerated approval trial. So they presented data on DLBCL and really a 3-month data on the first 51 patients of this 100-patient study. You're all aware of the disease, and this really included very chemo-refractory patients. So, and it's important to note that while these are chemo-refractory, I have to say that in putting patients on this trial, we noticed that these are still highly selected patients, because to get onto this trial, you had to be able to wait at least a couple weeks to go through this pheresis. And there's many patients with comorbidities or very rapidly progressive disease that you couldn't get onto the study. So I wouldn't say this is cherry-picked, but I would say that this is selected and may not represent, in fact, does not represent the bulk of the remaining problem of chemotherapy refractory large cell lymphoma. And there's 2 cohorts in the study, the DLBCLs and then the transformed folliculars, not Richter's, but transformed folliculars and primary mediastinals. And so they get enrolled in leukapheresis, and there is this conditioning chemotherapy, which is designed primarily to decrease the lymphocytes and to make immunologic space for the incoming T cells. But I have to say that this chemo is active. It's pretty intense chemo, and some of the activity, the early responses, say 1-month responses, I want to make a point, there is going to be some activity, this chemo, but it's not going to last because these patients have all been refractory to alkylating agents. And you can see that they've got 3-month follow-up on 51 DLBCLs. So look at the numbers. The response rates, indeed, at 3-month follow-up are very impressive. 79% objective responses, 52% CRs. However, at the 3-month assessment, you can see the CR rate is already falling. So there are patients that are relapsing, and we don't have PFS curves and that are mature. And it's really the 6-month data that the FDA has asked for in terms of any regulatory approval. So really, that's what we really need to wait for, and that'll be upcoming later this year. But this is still very, very impressive because these are really nasty patients. I know all the patients that we put on really were in big trouble, and there was really nothing else for them. And here's an example of a very robust response in a patient, chemotherapy refractory disease, and achieved, within 3 months, a really amazing response. Once again, this therapy, another point is to make is that this is very toxic therapy. There's a cytokine release syndrome. These can put patients in the ICU, grade 3 neurologic events. There were no cases of cerebral edema and deaths related to that, but there were 3 deaths in this 100 patients, 92 patients here, 2 related, and there was 1 that was a sepsis, and you could say that that might have been related to the incoming therapy. So this is something we have to consider when we talk about applying such a therapy to, say, patients with follicular lymphoma who may have other options. This is a dangerous therapy, so perhaps it's best to use for people who really have their backs against the wall. A lot of toxicities. Most people make it through. So if you notice here, here's the bottom line, is that we need to really see the long-term follow-up and what is going to be the progression-free survival in this group of patients. And we get some, like you see, 39% durable CRs at 3 months. We really want to know what is the shape of the PFS curve when we get out to 6-month minimum follow-up for these cases. And here's some data from the Seattle group, just to be fair, is from Juno, and they have a little bit different product where they give a mixture of CD8 and CD4, an equal defined ratio that they transduce with the CAR. And here's data in B-cell lymphomas. These are mostly DLBCLs. When they use the flu-cy conditioning, they get good response, but still the median PFS was only 5 months, even in the favorable group using the right conditioning regimen. So these treatments are not the answer to all of our problems. I'll just share with you briefly some different work that we're doing in our laboratory, and that is taking advantage of the multiple pleiotropic effects that interferons have. An old drug for lymphomas and cancer that has great activity against the tumor cells and has wonderful ability to activate almost all of our friends here, different immunologic effectors, but it's too toxic to administer. So what we've done is actually engineer an antibody. This is actually the V regions of rituximab with in a genetic fusion with type 1 interferon. We've made it with alpha and even beta interferon and shown that this fusion protein is more active at inhibiting lymphoma cell growth, and rituximab causes higher levels of apoptosis. And when you look at it in xenograft models, it's a very much more effective than rituximab. Here, this model is largely rituximab insensitive, and we see we're able to eradicate most of these tumors with this fusion protein. So based on this, just over a year ago, we started a phase 1 first-in-human trial. The FDA made us start with the smaller doses than we were giving to mice. So we're dose escalating very slow, and we're finally this month up to the dose that I think would be biologically and perhaps clinically active. So we're looking forward to that. So in conclusion, for immunotherapy, I'd say the anti-PD-1 antibodies, Hodgkin lymphoma, it's a no-brainer. It's an excellent response. It's approved as a single agent in this setting. Anti-PD-1 for B-cell lymphomas, responses are seen, but not likely to be valuable as single agents. You've seen some anecdotal exceptions to that. So we certainly want to try to build on these cases. I tried to look at and make a list of the combinations of PD-1 plus other agents. There's over 100 trials in B-cell lymphomas in clinicaltrials.gov of literally dozens and dozens of different agents in various combinations. So it's really daunting. CAR T cells, I think we have high response rates in underlying selected patients with chemotherapy refractory DLBCL, but there's also activity here, and it really remains to be seen what this is, what the durability of these responses are. And I asked the question, what PFS would make this therapy worthwhile given the expected high costs? I mean, these patients are refractory. We'd want to do it at any cost, but they're going to be charging a lot of money for this therapy. And really, what's going to be the benefit overall? Other novel therapies, we have so many to choose from now, and we really need to focus on these chemotherapy and rituximab refractory diseases. And I should end by saying that it's very difficult to predict which combinations are going to be most valuable, given that sort of animal models and in vitro models are very unpredictive. A big surprise from our study of nivolumab was that we saw no responses in multiple myeloma, but then pembrolizumab combined with lenalidomide in multiple myeloma had 50 to 76% responses. So that was kind of a really big surprise and shows that you can't always predict. Sometimes just combining things empirically that seems like a good idea, you can get some great results. So I'll stop there.